Now we are going to see about linkage. Introduction. Linkage is defined as the tendency of two or more genes to remain together in the original combination in the same chromosome during the process of inheritance for a number of generations. All the genes on a chromosome are said to be linked to one another. Linkage was discovered by T.H. Morgan, was found in linkage Drosophila, sweet peas, maize, man, etc. In Drosophila, the genes of body color and nature of wings are linked and located on the same chromosome. In sweet pea, the genes of flower, color and size of pollen grains are linked. In maize, the genes for color and shape of seeds are linked. In man, the genes for hemophilia and color blindness are linked. T.H. Morgan proposed the theory of linkage based on his experiments in Drosophila in the year 1911. The theory of linkage explains the main principle of linkage. Principles of linkage. A chromosome contains many genes. The genes are arranged in a linear fashion on the chromosomes. The genes present in a chromosome are linked. The linked genes are inherited together from the parents to the offspring. Linkage is a rare phenomenon when compared to crossing over. The strength of linkage depends on the distance between the linked genes. Closely located genes show strong linkage. Widely located genes show weak linkage. Mendel's law of independent assortment is not universal. The linked genes do not show independent assortment. The genes located on different chromosomes only show independent assortment. When dominant genes are located on one homologous chromosome and the recessive genes on the other homologous chromosome, the arrangement of the linked genes is called cis arrangement. When the dominant and recessive genes are linked in one chromosome, arrangement is called trans arrangement. All the linked genes of a homologous pair of chromosomes constitute a group called linkage group. The number of linkage groups in an orgasome to the number of pairs of chromosomes are equal. Thus, Drosophila has 4 linkage groups as it has 4 pairs of chromosomes. Pea plant has 7 linkage groups as it has 7 pairs of chromosomes. Man has 23 linkage groups as it has 23 pairs of chromosomes. Linkage is of two types, namely complete linkage and incomplete linkage. In complete linkage, the chromosomes do not break, and in incomplete linkage, the chromosome break occasionally and the linked gene inherit together for many generations. Example, F male Drosophila. Genes separate due to crossing over. Example, F female Drosophila. Arrangement of linked genes. Sturtevant in 1913 found out that the genes are arranged in the chromosome in a linear manner. Based upon the arrangement of the genes in the chromosome, the linkage can be classified into two types. Cis arrangement. In cis arrangement, the two dominant genes B and L are located on one member of the chromosome pair. The two recessive genes B and 7 are located on the other. This type of arrangement with two dominants on same chromosome is called cis arrangements. The heterozygotes with such arrangement are called cis heterozygotes. Trans arrangement. In this type, the dominant gene of one pair and the recessive gene of other pair are located on one chromosome, and the recessive gene of the first pair and dominant gene of the second pair are located in the second chromosome. The heterozygotes with such arrangement are called trans heterozygotes. Theories of linkage. There are two theories on linkage. First, differential multiplication theory, and second, chromosome theory of linkage. First, differential multiplication theory. Bateson 1930 assumed that linkage is due to differential multiplication of cells containing different combination of genes. After the segregation of genes during gamete formation, the set of gametes possessing parental combinations multiplies rapidly than the set having non-parental combination. This theory was disapproved because after segregation of genes there is only one division. Chromosome theory of linkage. Morgan and Castell proposed the chromosomal theory of linkage. The main features of the chromosomal theory of linkage are the following. The genes are arranged in a linear fashion on the chromosomes. The genes on a chromosome are linked. Genes showing linkage are located on the same chromosome. Linked genes will remain together during inheritance. The distance between the genes will determine the strength of linkage. The closely located genes show strong linkage. Distantly located genes show weak linkage. Importance of linkage. Linkage presents variation in evolution. Linked genes of desired traits help the plant and animal breeders in hybridization programs. Linkage groups. All the linked genes of a homologous pair of chromosomes form one group called linkage group. In Drosophila, there are 4 linkage groups corresponding to the 4 pairs of chromosomes. The garden pea plant has 7 pairs of chromosomes and has 7 linkage groups. Man has 23 linkage groups corresponding to 23 pair of chromosomes. Factors affecting linkage. Linkage is affected by the following factors. Distance. Closely located genes show strong linkage while genes widely located show weak linkage. Age. With increasing age, the strength of linkage decreases. Temperature. Increasing temperature decreases the strength of linkage. X-rays. X-ray treatment reduces the strength of linkage. Linkage in Drosophila. In Drosophila, grey color capital G is dominant over black color small g, and long wing L is dominant over vestigial wing 1. The genes for these two characters are linked together in the same chromosomes. The linked genotypes of the parents are written as follows. Grey long, capital G capital L by capital G capital L. Black vestigial, small g small g. When a fly with grey body and long wings, capital G capital G capital L is crossed with another by having black body and vestigial wings, small g and small f. All the capital F individuals are having wry body and long wings. When F male hybrid is test crossed with the double recessive parent, the F contains only two types of individuals in equal numbers instead of expected 1:1:1:1 dihybrid test cross ratio. In the F generation, all the flies are like their parents. There are no new combinations. This is because the gene capital G is linked with capital L in one chromosome and the genes small g and small l are linked in another chromosome. Hence, during gamete formation, they cannot segregate independently. Hence, only two types of game are formed, resulting in the formation of two types of combinations in capital F. The resulting offspring are resembling the grandparents. This is because gene capital G is linked with capital L and the gene small g is linked with capital L. Types of linkage. Linkage is of two types, namely complete linkage and incomplete linkage. Complete linkage. In complete linkage, linked genes inherit together for many generations. Here, crossing over does not occur. In complete linkage, the genes are closely situated. Body color and shape of wings in male Drosophila. Second, bent wings and shaven bristles in the fourth chromosome of Drosophila. Complete linkage is the phenomenon in which two or more genes or characters are inherited together for a number of generations. In this, genes are closely associated and tend to inherit together. Complete linkage is due to the fact that there occurs no break in the chromosomes. As a result of complete linkage, the young ones inherit only the parental characters. New characters do not appear among the young ones. So, complete linkage produces only parental combination. New combinations do not arise. This phenomenon is very rare. It is found only in male Drosophila. The F male hybrid is back-crossed with recessive female parent. The F male hybrid produces only two types of gametes in which the linked genes are inherited together. So, only two types of offspring are produced in the F generation in equal numbers. Incomplete linkage. The separation of linked genes during inheritance is called incomplete linkage. The linked genes are separated due to crossing over, chromosomal breaks, etc. Example: body color and wing shape in female Drosophila, seed color and seed shape in maize, flower color and pollen grain shape in sweet pea. In incomplete linkage, the linked genes on certain occasions separate. This leads to the formation of new combinations among the young ones. Incomplete linkage is due to the breakage of chromosomes during gametogenesis. Incomplete linkage is found in female Drosophila. This breakage of chromosomes leads to the separation of linked genes and new combinations appear. Because of this new genic combination, the offspring produced in the F generation are differ from their parent in their phenotype and genotype. So, the incomplete linkage involves the accidental breakage of chromosomal segments or linked gene resulting in some combinations of genes. Thank you.